Okay, I think it's time to get going. So thank you for joining us. I'm Rex Malmstrom. I'm a scientist at JGI, and I'll be hosting today's meeting where we discuss the JGI's New Investigator Award, or New Investigator Call for proposals. And I hope this presentation will clarify some of the points of the application process. And we also hope it will provide an opportunity to address your questions about the call. The questions will be collected through the chat bar, so please send those as we go, and we'll take some time at the end of our discussion to address those questions. Before that though, a little bit about us. The Joint Genome Institute, or JGI, is a US Department of Energy user facility, and we're located at the Lawrence Berkeley National Laboratory in the Bay Area in San Francisco. We're world leaders in the genomics of plants, fungi, algae, and complex environmental microbiomes. And we help researchers characterize how these different organisms and how these different communities carry out complex biogeochemical processes, as well as how these organisms can be harnessed for sustainable biofuels and bioproducts. We have resources in the area of DNA and RNA sequencing, DNA synthesis, metabolomics, data analysis, and these capabilities are provided to researchers for free based on a competitive peer-reviewed process through our Community Science Program, or CSP. And today's presentation is going to focus on the New Investigator Call, which we typically have twice each year. I'm going to point out though that the submission deadline for the next New Investigator Call is coming up quick. It's September 21st, so it's about 3 weeks away. To better explain how this New Investigator Proposal works, we'll be hearing today from Krista Pinocchio, head of the JGI's project management team, and Tanya Wojcicki, director of JGI user programs. One of the common themes that you'll notice in today's presentation is that it's a good idea to contact us if you have questions about your proposal, whether it would be a good fit in terms of our capabilities, the DOE mission, and whatever else it might be. So that's a common theme. Please reach out to us when you have questions. And speaking of questions, again, I encourage you to submit questions via the chat bar, and we'll do our best to address them in this meeting. Okay, so we'll get started here. I have a question for Tanya. Tanya, who is eligible to become a new investigator?
Yeah, David, maybe you can skip to the next slide. All right, so as you can see on this map, this is our FY19 user map. We have users from all around the globe. In that set, this particular call is open to both domestic as well as international applicants from pretty much any institution, as well as any type, any career stage. This also includes postdocs. So we're really trying to draw in young investigators. For the new investigator call, however, as the name alludes to, there's one criterion, which is that the applicant cannot have been a lead PI on a previous proposal accepted by the JGI. So the purpose of this call is really, this call is specifically intended to bring in new users to the JGI.
Great, thanks, Tanya. Krista, could you tell us a bit about the types of services and capabilities that can be requested through the new investigator call?
Sure. So if you could get the next slide, please. For each of the calls that we offer, we have different allowable products, and the new investigator call currently includes things like de novo genomes, and so this could be either from bacteria, archaea, or sorted cells. We also have sorting capabilities. And the standard analysis for this kind of product would include an assembly and an annotation. We allow genome size evaluations for really large genomes like algal and plant. These are specifically targeted toward providing pilot level data on genome size and genome complexity, and more for preparation of a future large scale proposal submission. We offer resequencing from all of the key target organisms. And this kind of project relies on having a already closely related reference genome that has been sequenced and annotated. And so for these, the analysis will include mapping raw reads to the reference genome and identifying differences from single point mutations to structural variations that are larger. Gene expression, or RNA-seq, is also offered. We can prepare either polyadenylated selected libraries or ribosomal depleted libraries. And for analysis, we do mapping of those reads to the reference genome, and then giving you the raw and normalized counts for each of the putative transcripts. The metagenomes and metatranscriptomes are for sequencing of communities, so environmental samples, and the analysis will include assembly and annotation of the reads. Metabolomics includes analysis of both polar, such as amino acids or nucleosides, or non-polar metabolites, such as lipids and secondary metabolites. So we use liquid chromatography combined with tandem mass spectroscopy. The output from metabolomics products is metabolite annotation, and this is based on standards that we've run here at JGI or predicted using computational methods. We can also do relative abundance prediction for the identified metabolites. And then finally, for synthesis, we offer synthesis of single genes, gene clusters, and pathways, and these can be used for functional characterization. We can do synthesis of combinatorial libraries for metabolic pathway engineering. And we also do complex libraries such as sgRNA libraries for use in CRISPR-Cas9 technologies. And for those, we provide constructs that are cloned into the vector of your choice. And I just want to note that for all the sequencing metabolomics projects, the raw data is also provided, so you can do additional analysis within your own team. So JGI is not a funding agency. We don't have a budget associated with these proposals. Users are expected to provide the DNA or RNA or other samples, and JGI does the library prep, sequencing, or metabolomics and synthesis and basic analysis.
Great, thanks, Krista. Now that we had a bit of an overview of what can be requested in the proposal, could you tell us a bit more about how to submit a proposal and the format of that proposal?
Yep, and this is one of the most commonly asked questions that we get. Could we get the next slide, please? So in the red circle, you'll see the URL where you can submit a proposal, and there's a red arrow next to the CSP New Investigator Begin Proposal link. So the New Investigator proposals follow a very short web-based format. It's almost like a white paper format. And you can view the template by creating a JGI account and starting a new proposal using that the link by the red arrow. So the JGI account will take about 2 or 3 minutes for you to create, and it gathers some basic information that our DOE sponsors ask us to collect about our users. If we could get the next slide. Once you start the proposal, the top part of the proposal form includes all the different products that are included in this call, the ones that I just went over. And it also has a number of samples that are allowable for each of the product categories. In addition to determining the number of samples that you are requesting for each of the products, you also need to estimate the total amount of sequencing that would be required. And this is to make sure that you stay within the 500 gigabase sequence allocation that we have on this call. So there's a link provided in this section. It's circled here with a red circle. And it gives a little more information about the amount of sequencing that's typically applied to each of these products, and this will help you with your calculations. It's not a one-size-fits-all, and there can be variability depending on complexity of your environmental samples or size of your genome, especially for eukaryotic genomes. And so this is something that we'll give you hints on, and if you have questions about it, we're more than happy to discuss with you. It's important to not request too much over the 500 gigabase cap, because that will lead to issues during the scientific review. Can we get the next slide? The next section of the proposal form is sort of the heart of the proposal. There's 6 sections. The first 3 are shown here. Each of these has a 4,000 character limit, which corresponds to roughly 500 to 800 words, so a paragraph or 2. Don't feel like you have to use the whole thing for each of these sections, but especially for the description and justification sections, you'll probably want to use most of it. So I'll start with the description. In this section, we're looking for background on your study, what scientific questions you want to address, describe your experimental design, what's your study site, for instance, and what are you requesting in terms of sequencing, synthesis, and metabolomics. For the justification section, this is a place to highlight the importance of your proposed work. Briefly summarize the research field and how your work will advance it. And remember that the scientific reviewers, while they are experts in genomics, They may or may not be experts in your specific system. So make sure they have the background and they can see where your proposed work is going to fit into the scientific field. And then for utilization, this is a place to describe your plan for analysis. So JGI will give you a standard output for each of the products, but what will you do with that data once you have it? Are there additional analyses that you plan? What resources and tools are you going to use to do those? And this is a great place to highlight your team's expertise in the area, or how you might build that expertise if you don't already have it. Okay, the next slide will continue the next three sections of the proposal. Community interest section is designed to highlight the impact or benefit this data will have for the scientific community as a whole. So think about how the data will be used by other researchers, not just your own group. And remember that sequence data have very long lives after initial publication. So they're often used again and again by different researchers. One good way to demonstrate community support is to have letters of support from investigators who will be using your data, and they can specifically say what about that. What about the data they will use in their own work? DOE mission relevance is an important section. Because JGI is a DOE user facility, it's important that the work that we do is relevant to the DOE mission areas of interest. And so we're going to talk about this next because it is, we do need to provide a little more detail here. And then the final section is sample preparation. And this section addresses your strategies for preparing DNA or RNA or other materials that you'll need. Can you get sufficient quality or quantity of material? Are there any technical difficulties you're anticipating, and how will you overcome them? When will the samples be ready to ship to JGI? It's not required to have the samples in hand, but it definitely helps if you've been able to demonstrate that you can extract sufficient material from similar samples. And note that JGI does not do sample collection. We don't fund the sample collection, and we don't do the DNA or RNA extraction. So that's something that will be done in your own lab.
Great, thank you, Krista. Tanya, could you please speak a little bit more about the DOE mission and DOE relevance?
Yeah, definitely. David, do you mind moving forward? So the DOE's interest as it specifically pertains to the JGI are in the area of bioenergy, bioproducts, and biogeochemistry of different systems. This means that the mission is fairly broad. We don't take projects that are focused on biomedicine, human health, or food crops. For example, studies about human microbiomes or human pathogens would not be appropriate. However, many other microbiomes that are tied to geochemistry or bioenergy would be. There's some overlap between some food crops and bioenergy crops, so that means that we have a bit of a gray area there. The same is true with studying some plant pathogens. Generally, it's a good idea to seek clarification if the system being studied would also seem a good fit for NIH or USDA funding. This is also true for studies of marine systems, since DOE Biogeochemistry mission is heavily focused on terrestrial ecosystems. Studies in coastal waters are specifically identified as fitting the DOE mission, but projects focused on biogeochemistry in the open ocean are not, unless they focus on either basic science that sort of extends beyond the marine realm, or they develop methods that can be translated to the terrestrial ecosystems. You can find more details on our website, but if you have any questions or if you want to get more guidance, always come talk to us and we can discuss your particular project and its DOE relevance together.
Thanks, Tanya. Krista, are there some additional components for reviewing proposals that are requesting DNA synthesis?
Yeah, that's a good question. So in the proposal form, if you do indicate that you'll be requesting DNA synthesis, you'll get an extra section to fill out, and some of it is shown here in this screenshot. All proposals that request synthesis go through an additional review panel, and their job is to evaluate the biosafety, biosecurity, and any environmental aspects of the proposed work. So for proposals that request DNA synthesis, there are a few additional pieces of information required. So there's a series of yes or no questions shown up at the top. And these are mainly related to pathogenicity, antibiotic resistance, toxicity, and that sort of thing. Followed by the screening section, which is that the second comment box in the screenshot. So in this section, you should address some possible risks associated with the work and offer strategies to mitigate those risks. If the committee doesn't feel that sufficient thought has been put into the response, for example, if you just put, there are no risks associated with this work, the proposal won't be approved unless modifications are made. So we encourage you to think broadly and think long-term about possible impacts of this work.
All right, great, thanks, Krista. Tanya, can you tell us a bit more about how proposals are reviewed?
Yeah, definitely. So there are two components to a proposal review. First of all, there's a technical review, and then second of all, there's for the technical review, these are performed by JGI staff. In those, we want to make sure that the proposals are aligned with our in-house capabilities, with the scope and the scale of the call. For example, if the sequencing request is 10-fold higher than our sequencing CAP4 call, that would raise a red flag. So we'd have to talk to the PI. For the annual, both our annual CSP call and our FICOS calls, there is a letter of intent stage, which generally gives JGI the opportunity and helps JGI to provide feedback to the PI prior to the full proposal submission and ensure that the scope, the scale, and the DOE mission relevance are on track. For the new investigator proposals, however, there's no letter of intent stage. So you're submitting the full proposal right away, which means it's a really good idea to contact JGI staff for feedback before you submit the proposal to just make sure everything's on track. Then second, for the scientific review, we do rely on a panel of scientific experts. These are external. They review and rank these proposals. JGI scientists do not participate in the scientific review or the rankings. And if we look at the list of criteria that are most important for the ranking, is the really most important point is the scientific merit or the scientific excellence. Other criteria include the DOE mission relevance and as well as the proposer track record and ability to utilize the data. We want to make sure that if we give you these different data sets, and often it's a quite sizable amount of data, that you will be able to use it, you will be able to analyze it and use this data to meet your goals. So that so then the next criterion is the value of the data to the user community. And here we want to make sure that the data is being used more broadly, ideally not just by a single PI, but that the data is of use to other researchers and a larger research community. And then lastly, the reviewers want to make sure that the PI has a high chance of attaining the stated goal. Again, with the data that we give you, are you able to answer your scientific questions? Once we have the scientific review and the ranking of the proposals, this list will go to the Department of Energy, and DOE will review these ranked proposals. In some rare cases, they may suggest removing a proposal if they feel that it doesn't align with the DOE mission. While this is rare, again, it can be avoided generally if you discuss your proposed work with JGI beforehand, so we can help you.
Thanks, Tanya. Can you tell us a little bit about the success rate of these proposals?
Yeah, that's a great question. So it's pretty good in comparison to most funding agencies. Depending on the call, the success rate are typically in the double digits and most often greater than 25%, sometimes even around 30%. And this is in comparison to the single-digit success rates of many other funding agencies. It's fairly high. Again, a successful proposal, and Krista mentioned that also earlier a couple times, is granted access to capabilities and services and not actual funding. So we will not provide funds for you to collect the samples or to extract the nucleic acids and carry out your proposed experiments or prepare your results for publication. We only provide the services of the sequencing or metabolomics. We do post all successful proposals on our website. Actually, David, if you can skip to the next slide. And this is the link right here, or the website. That gives you an idea. You can have a look at what other people have proposed. A high-level description of their project is listed on the website. So that gives you a feel for, over the years, what are the type of projects that have been accepted and that we are working on.
Great, thank you. You know, one question we often get is about the data release policy and data usage. Could you please tell us a little bit more about that?
Yeah, definitely. David, if you want to skip on to the next slide. So all sequence data will be released in accordance to our JGI data release policy. Data from all CSP projects, again here including the new investigator call, is eligible for immediate public release to the JGI genome portal. There are some products, which is the standard metagenome and metatranscriptomes. For those, the analysis files will be made public on the portal 60 days following the project completion. And these 60 days are really there to allow the PI to look at the data more thoroughly, to do a brief QC, sort of we call this a review period. We just want the opportunity to do a sanity check of the data before it goes public. All other data files, again, are made immediately available on these project portal pages, and this is the DOE mandate. Now, one thing to point out is that there are restrictions for other scientists to use a PI's data, so there is a level of data protection. Scientists are expected to contact the data owner about their intention to include a PI's data set in their analyses or publication, just to ensure that their publication does not compete directly with planned publications, or what we call reserved analyses of the principal investigator. Now, these restrictions will be lifted upon the publication of the data set by the data owner, or 2 years after the public release of the data, and whichever comes first. All of our policies are listed in detail on the JGI website. And again, if you have questions, you can ask us for more details.
Great, thank you, Tanya. As one last question, at least in this prepared section, and it is if you can tell us what are some of the best indicators of success for a new investigator proposal.
Yeah, so first and foremost, carefully read our proposal call description and ensure your idea is aligned with everything. Discuss your proposal idea with JGI staff to clarify any points about JGI capabilities, the technical feasibility of your work, the relevance to the DOE mission. Again, as there is no letter of intent stage for the call, this is your opportunity to make sure that the proposal is going to be appropriate, and ideally do that more than 1 day before the deadline so we have time to respond to you. Have someone proofread your proposal. Make sure your goals, experimental design, your plan, data utilization are clearly communicated in your proposal. And lastly, and this is really important, describe in detail what you will do with the data. This is often the weakest area of a proposal, not just the new investigator proposal, but CSP proposals in general. Ideally, you or someone on your team will have experience in this area, and you can demonstrate in your proposal how competencies can be built. You can show and communicate what you're going to do with the data that JGI will give you. But again, importantly, come talk to us with questions. David, if you want to skip to the next slide. We have a team of experts at JGI that are focused in specific scientific areas or technical areas. There's just sort of a screenshot of various scientists that are often responsible for answering your questions. So we will farm out these questions depending on what they are and which focus area they are. And we are here to help you.
Great, thank you. I think this is all hopefully useful information for the folks who are attending online. I would like to remind the folks who are online that there's an opportunity to answer any questions that you have. I've gotten a few of them here, so we'll just get going answering some of these submitted questions. So Krista, one of the questions is asking whether or not people can mix and match options in the proposal as long as they're staying under the sequencing cap. Like, can they request metabolomics and resequencing?
Yeah, that's a great question. I should have addressed that when I was discussing it. Certainly and definitely we encourage this. There's multiple options, ways you can mix and match synthesis, metabolomics, sequencing. Yeah, the only requirements are that you stay under the 500 gigabase cap and that you don't request more than the specific sample outside of the sample range that's listed there. But certainly proposals that are looking at multiple areas or multiple ways to answer questions are definitely encouraged.
Yes, thanks. I have a follow-up question for you, Krista, also about this. There's a question asking if you could speak about the pros and cons of proposing a truly exploratory small proposal that might not max out the whole 500 gigabases. Or is it better to, try to reach that maximum with broader questions and broader project scope?
Well, I will say that we are looking for projects that can't easily be done at a core facility. So if you're looking for 1 or 2 microbial drafts, I would say that's probably not a great fit. But I don't think you need to necessarily just add samples in order to max out the gigabase allocation. I think strong science and well thought out questions, whether it's kind of a complete standalone study, which is a little more unusual for this call, or more commonly getting pilot data prepared for a larger proposal submission in the future. I think, the scientific review criteria that Tanya went over are definitely how we're going to be judging these, regardless of the size. Tanya, do you have anything to add to that?
Yeah, I can complement that a little bit. So at least I completely agree with everything you said. And for some of the products, we do have minimum requirements. So, and that is a little bit depending on how we batch things and how we run things in a production setting. So as Krista mentioned, for microbial isolates, we're not going to be able to sequence just 1 or 2 genomes if somebody sends us 1 or 2 tubes. So we have minimum requirements. They're listed on the website, for example, at least 24 microbial genomes. So yeah, these details are available on the website.
Great. I have another question here asking that, well, they are a PhD student and they're asking if they would be allowed to submit a proposal, including maybe their advisor as a co-PI, but can PhD candidates be considered for the new investigator proposal?
Yeah, I can answer that one. Yes, I think at most institutions, a PhD student can be considered a PI. There's no restriction from JGI's side, but do check with your institution as there are some that won't allow this. But I do think it's really important to show that your team has the necessary experience to handle the data, either by adding team members as co-PIs or collaborators on the proposal, or at the very least providing letters of support saying that they will support this work.
Okay. I have another question here. This one is, I think this would be a question for you, Tanya. Someone was asking that they had submitted a proposal previously. They got good reviews, but there was questions about their ability to analyze the data, and we're looking for advice on how that they could better demonstrate their capability for being able to carry out the work and utilize the data they were requesting.
Yeah, that's a really good question. So the reviewers do look at the PI's sort of track record. They want to make sure that that this PI has already worked with this type of data set. I think having publications in this area would be helpful, just to show that you've worked with this type of data set, you've analyzed it, and you've published on it. Alternatively, a lot of our proposals have co-PIs. So we have PIs that may have never worked with genomic data sets, yet they're requesting large amount of sequence data from genomes. But then they have a co-PI who has extensive experience in bioinformatics and analyzing genomes, and teaming up with them can also make a really strong proposal. So you kind of have 2 PIs that have complementary skills that can make a really strong research team. And that then lets the reviewers know that these PIs are really able to take advantage of the data and work with the data.
Yeah, that's great. There's a couple different ways to go there, and thanks for providing those suggestions. I'm seeing another question here. I'm happy to just answer this one. Someone was asking if they could contact the JGI prior to submitting to discuss the relevance to the DOE mission, and the answer is absolutely. And you can contact us not just about DOE relevance, but there are questions of whether or not what you're asking fits within our capabilities. We're here to help, and we want to, especially for the new investigator where we don't have a letter of intent stage, this is a chance for us to give a little bit of feedback to make sure that that the proposal is aligned with our capabilities and our mission. So we're happy to do that. Please reach out to us and we'll make sure to get back to you. Okay. All right. I have a question here, I think for you, Tanya, as well. Can you tell us a little bit about the analysis capabilities that are available for JGI?
Yeah, that's also a great question. So one point to emphasize, we didn't really do that yet, is that we're not just going to give you raw data. We will analyze the data, we will QC the data, we will, depending on the product, assemble it or do mapping. We have pretty extensive pipelines that are state of the art. We update them and keep them really up to date. And we have various systems which enable you to do comparative genomic analysis. So for example, for microbial genomes, single cell genomes, including single cell genomes and community sequence data, we have a system that's called IMG, the Integrated Microbial Genome System. That's a comparative genomic analysis platform in which we integrate your metagenome data set or your isolate or single cell genomes. And it allows you, it has a graphical user interface that allows you to analyze your data in the context of other publicly available data. It's a really great system. We also have courses. They used to be in person. And now there's a webinar series, and hopefully we'll come back to in-person training sessions that are open to anyone to participate and learn how to use these. And likewise, we have analog systems for our other organisms. We have a fungal system, microcosm and phytosomes for plants, where you can analyze your plant and fungal genome data, and now also an algal database. So there's quite extensive pipelines and tools that are available to you.
Great, thank you, Tanya. Krista, I have a question here about references in the proposal. Should they be included? Is there a section, or how would you include a reference or citation in the proposal?
There's not a specific section for references, but certainly if you're presenting preliminary data that's been published, or if you want to give some background of the field, references would be encouraged. And you can either put those in the individual text section if you have space, or you can upload everything as a supplementary file. There is a place to do that. PIs will upload their CVs as supplementary files. You can also upload other figures or reference lists there.
Great, thank you. Okay, I have one last question here for you, unless anything else comes in on the chat bar. But the last question I think would be for Tanya, and this is actually about sorting capabilities. The JGI does some preparatory work. Can you just tell a little bit about what can be requested in that area?
Yeah, absolutely. So JGI has a sorting pipeline, flow cytometry pipeline for sorting environmental samples. And in fact, that is a pipeline that Rex and his team are running. And it's a really great and I think very unique capability. So what PIs can do is they can send environmental samples. Ideally, those are cryopreserved to keep the cells intact in a glycine TE buffer or alike. So you send this cryopreserved material to Rex's team, and they will use flow cytometry to sort individual cells into 384 watt plates. These are then being lysed and whole genome amplified. And these whole genome amplification products can then be sequenced. We don't do 16S screening anymore of these, so you kind of get any single cell that amplified and can be sequenced. It's a really great tool. In addition to getting the sorting done at JGI, you could also send us the ACS if you have your own facility. And then we would create the libraries and sequence these single amplified genomes.
Great, thank you, Tanya. I haven't seen any more questions come in on the chat bar, so I think that means that it's time to just say thank you. So thanks, Krista. Thanks, Tanya, for explaining this thing. Thanks to David, Massey, and Marilyn, who are behind the scenes helping us with this presentation. And thanks to all those who joined in today and participated. If you still have questions and they weren't addressed, just send them. Send those questions to us. Reach out to JGI. We're happy to do that. We're going to take this video and a recording of this video and post it online. There'll also be links to other, we have a sort of general JGI FAQ section. A lot of information is online, and so I would urge you to look there. Please remember that the deadline for the next phase of the New Investigator Call is in about 3 weeks, so September 21st. But we also do the New Investigator Call twice a year, so there'll be another opportunity if you're not quite ready this time. You can stay in touch with the JGI and the latest information by following us on any of the various media platforms of Twitter and LinkedIn, Facebook. And I would actually like to throw a little shout out to the JGI podcast, the Genome Insider and the Natural Podcast, is that what it's called? Anyway, the two great podcasts. I listen to these when I'm out walking my dog in the morning. I work at JGI and I really enjoy these podcasts, so check them out. Okay, that does it for today. I hope the info that we provided was helpful. Good luck crafting your proposals on this call or on future calls. And we look forward to reading them when they come in. Best of luck. Thank you.